This transplanted Canadian has a PhD in chemical engineering. He's now a professor at NC State. He's been director of the Bioprocessing Science Program since it started in 2007. He founded the Wolfpack Brewery. He teaches manufacturing technology related to fermentation and biopharmaceuticals. Raise your beer glasses. Let's welcome Dr. John Sheppard.
Okay. Well, thank you, Andrea, for the nice introduction, and thanks to Alicia, I don't know where she is, for inviting me to speak here. I did speak here actually about 6 years ago, and it was 20 minutes, I think, unless my memory is gone. But anyway, so I'm going to try and get through this 5 minutes, and it's going to happen no matter what. So let me see. Okay. Here we are. Yeast, the magic microbe. So before I talk about yeast and why it's so magical, I'm going to talk a little bit about the perspective I have on yeast and how I got to know this unique microorganism. So, as Andrea mentioned, I'm a professor of bioprocessing science at NC State, and a lot of people don't know what bioprocessing is, especially outside of RTP. Oops, is it still going? Yeah. Okay, so what is bioprocessing? Well, essentially it's the use of microorganisms, single cells, also animal cells and plant cells for making new products. Okay, so this is done in bioreactors. As you can see, this is a 30-liter bioreactor we have at NC State, actually, we use for training purposes. Okay, so what is my relationship with yeast? Well, as Andrea mentioned, I'm a transplanted Canadian, so I first started working with yeast in the National Energy Program in Canada and also at McGill University in Montreal, and I was working on biofuels. So yeast are essentially the major, if not the only organism, essentially, making bioethanol. And that's where I first started my research with yeast. When I came to North Carolina, actually, though, I was hired to work at BTEC, the Biomanufacturing Training and Education Center. And there, yeast is more important for making biopharmaceuticals. Okay, so they're responsible for about 15% of the biopharmaceutical manufacturing. Other cells, E. coli and CHO cells, are also very important, but yeast is one of the key microbes. While I'm at NC State, I've been housed in the food science and nutrition department, as well as running bioprocessing, and I initiated the Wolfpack Brewery, as you can see, so I have a relationship with yeast with regards to making beer as well. Yeah. Okay, so what about yeast? Why are they magic? Well, it was about 200 million years ago when a change happened with the yeast. And what happened? Well, yeast developed the ability to make ethanol. Ethanol, obviously, is a very interesting molecule. We've all had some experience with it, I'm sure. And the thing about ethanol is that it gave the yeast an advantage over the other microbes in its environment. Ethanol is very toxic to most bacteria. And so the fact that yeast was able to excrete this ethanol into the environment gave it a competitive advantage over the microbes millions and millions of years ago. And so this ability to make ethanol actually has lasted a long time. Before we could get beer, though, we needed to have people. Remember, 200 million years ago, no one was around. Okay, so the yeast had to go on its own and essentially just behave itself in the environment for a long time before people arrived on the scene and decided that maybe we could use this yeast for some practical purpose. So civilization occurred. And with civilization, we got agriculture. And with agriculture, we got domestication of animals as one of the things that we had. And as you know, with domestication, what we did was we took wild animals and we made them more amenable to staying on the farm so that they would produce food for our communities that were no longer hunter-gatherer communities. And this domestication of animals improved the quality of the products from the animals. Well, actually, the same thing happened with yeast, but it was kind of a magic process with yeast because nobody knew what yeast was. Back 500-600 years ago, there was no microscope. Microscope came a little bit later than that. So when they were making beer, and certainly 500-600 years ago, there was a lot of breweries around in Europe especially. They've been making beer for about 3,000 years. So when the yeast came on the scene, they had to improve the ability, they had to domesticate the yeast. Well, they didn't know what they were doing, as it turns out. It just happened naturally. And how did it happen? Well, it happened because they were using open fermenters. The yeast was just falling into the fermenters, and they were making any kind of beer that actually happened to result. So how did they domesticate? Well, it was a natural process where they were recycling the yeast from one batch of beer into the other. And eventually, over many thousands of generations of yeast, this yeast became perfectly adapted to making good tasting beer. And that's how essentially the process of yeast domestication happened. It was a selection process that the brewers unknowingly did without even realizing actually that it was a microorganism that was responsible for all this. Okay, so this is a picture of a brewery in the Czech Republic that I visited, and that 1286 is actually not the address, but that's the year of incorporation of this brewery. So this is a pretty old brewery, and they've been practicing cold fermentation for many, many years, about 800 years in fact. And it was this specific environment in the brewery that actually resulted in the development of a special kind of yeast, which is the yeast that makes lager beer. So I don't know how many of you know much about making beer, but lager beer is made using cold fermentation. Okay, and this divergence of the yeast happened about 500 to 600 years ago when the normal ale, Saccharomyces cerevisiae, which is the ale yeast, is a warm-loving yeast, diverged, and now we have the cold-loving yeast, or cryo-tolerant yeast, as it's called, for making our lager beer. Okay, so how important is yeast from a commercial point of view now? Well, you can see here I've shown the major products from yeast. Beer is worldwide the most valuable product, about $610 billion annual. Bread, and then wine, and then we have a little gas pump there that going back to my biofuels days. So the bioethanol is worth about $40 billion a year. And then at the end of the line here, we have biopharmaceuticals. So yeast actually is one of the major insulin makers. So recombinant insulin is manufactured by yeast as well. Okay, so what's the future of biomanufacturing? Well, essentially, remember, we're using these microbial cells to make these high-value products. Well, how can we tweak these cells in order to make the products that we want? Well, we can use genetic modification. So things like CRISPR technology can be used in order to modify the cells in order to make some of these high-valued products. We can also use classical methods, which is strain selection, crossbreeding, etc. But probably the newest or hottest thing on the block right now is what's called bioprospecting. And bioprospecting means that we're going back into the environment and we're trying to isolate new species or new strains of microbes that will give us new products. And the whole idea here is then we select samples from nature. I've shown here some bees. One of the products I've been involved in developing was a yeast species that we actually isolated from the gut of a bee. So you isolate those cultures, you purify them to get rid of any competing microbes, and then you go through a laboratory and commercial procedure of domesticating so that you can get a new higher valued product. And there's actually unlimited numbers. There's tens of thousands of microbes in the environment that we've yet to discover. And all of those microbes have individual unique capabilities for making new products. So thank you for your attention.
Thank you so much. Questions for Dr. Sheppard? Any questions? We'll go back over here.
Awesome stuff. I mean, you know, some of us maybe like to drink a little beer. Yeah, so my question is, this is fascinating in terms of where we are today. What are some of the examples that you can think of to where this is going? I mean, you mentioned new products. What can we, you know, eagerly anticipate? And thank you again.
Okay, so depends on what product category you're talking about. If you're talking about beer, maybe you did. Okay, if talking about beer. One of the important things about yeast and the fermentation of beer are the flavor components that the yeast manufacture. These are, a lot of times, they're esters that give the fruitiness and aroma to the beer. And a lot of the wild yeast are making different compounds than these domesticated yeasts do. The problem with domestication, well, it's good and it's bad. Domestication gives us a very high yield of ethanol, which is something the brewers want, obviously. They want good sugar conversion efficiency, so there's not a lot of sugar left over after the beer has been fermented. But the problem with some of the domesticated yeast is they don't give the kind of flavor variety that used to be back in the old days when they had every brewery had their own yeast that they collected locally or that was floating around inside the brewery and fell into the fermenter. So we've lost a lot of variety in the product as a result of that. So this bioprospecting technique allows us to essentially go back to the 19th and 18th century kind of beers and with the improvement that we're going to have more ethanol, but also we're going to have more flavor variety. So hope that kind of answers that. I don't know.
You got me pumped up.
Okay.
Questions somewhere, right?
Hey, I'm also an NC State PhD student, but I was wondering about the bees and their, you said you were gathering some of this yeast from the gut of the bees,
Yeah.
but how about the carbicula, like the basket of the pollen is collected with the bees? Do you know the term of the?
What was the question again? Sorry?
My question is the
Yeah.
carbicula that you can also get the yeast from the wind pollination, that all these pollens are getting together in the carbicula of the bees. So I was wondering if there's any way to get the yeast out of these carbiculas.
To isolate the yeast from the pollen? Is that what you're saying?
Yes.
Yeah. So yeast is pretty well ubiquitous. I mean, it does have favorite environmental niches that you find larger populations and more variety in, but you can isolate yeast from almost any natural source in the environment. So pollen would be potentially a good source as well.
We'll go back here.
It's very interesting to learn that yeast can produce medicine. Are there certain types of molecules yeast are optimized to produce?
Yeah, good question. So yeast is a eukaryotic organism, unlike E. coli, the bacterium. And because yeast is eukaryotic, it has certain innate abilities to make complex proteins, like human proteins, and to so-called post-translational modification of the proteins. So the yeast is very good for making complex proteins like insulin without requiring any additional chemical modification to make them suitable for use as a therapeutic protein in humans. Okay, so they can actually make human proteins essentially identical to what our own body makes. Yeah.
Thank you so much. Let's hear it for Dr. John Sheppard.
Thank you.